a master's degree. Angelique finished her veterinary degree in 2019 at Onderstepoort and went on to do a master's in tropical animal health, which she obtained this year. She is currently living in Australia, and her interests are zoonosis and the transfer of that. The floor is yours, Angelique.
Thanks, everyone. I hope you can hear me okay. I'm just going to start sharing now.
We can hear you, Angelique.
Can you see my presentation?
Yes.
Yes, okay, perfect. All right, thank you everyone. My name is Angelique Jacobs and I'll be presenting for you today on my project titled Investigating Rodents and Environmental Samples as Possible Sources of Leptospira Infections in the Mnisi Communal Area, Bushbuckridge, South Africa. It's quite a mouthful, so do bear with me. So leptospirosis is a zoonotic bacterial disease not only causing economic losses in livestock farming, but it also has a high morbidity and mortality in humans. Leptospirosis has a worldwide distribution but remains underdiagnosed in Africa due to the lack of resources and the presence of other febrile illnesses such as malaria. Leptospirosis is caused by Leptospira species, aerobic bacteria that survive optimally in warmer, more humid climates and is prevalent in tropical and subtropical regions. Leptospires are able to survive in soil, contaminated water, animal or human hosts. Leptospirosis has been regularly researched in the veterinary setting as it causes more severe economic losses in the farming industry by means of abortions, stillbirths, decreased milk production, and even death. Humans can also be infected, and it is therefore classified as a zoonotic disease, leading to morbidity and mortality in humans. The infection is carried and maintained in the renal tubules as a chronic renal infection, and sometimes in the genital tract by numerous wild and domestic hosts. These carrier animals excrete leptospires intermittently or continuously in their urine, contaminating the environment. In most tropical areas, rodents are hypothesized to be the most important reservoir host leading to human infection. Taking this into consideration, there is an increased risk in areas dominated by poor hygiene, urban slums, poor sewerage, and regular floods, as this may expose humans to rodent urine. Leptospira species has historically been classified into pathogenic and non-pathogenic species. It is directly spread through infected urine and indirectly by contaminated water. The bacteria gain entry by means of skin abrasions or cuts or through the nasal, oral, ocular mucous membranes. Virtually all mammalian species can be infected with Leptospira. Some act as maintenance hosts, others as incidental hosts, and numerous species of Leptospira may incur a host intermediate state. Maintenance hosts differ between the various Leptospira serovars, but some of the most commonly described hosts, as depicted in the Merck Veterinary Manual 11th edition, is set out in the table below. I'll just pause for a second to have a look at that. All right, so I added this very basic slide in to show the amount of literature available within the Southern African development community. These very basic images were prepared by me during preparation of my study to visually depict the lack of research on leptospirosis within Southern Africa. There may be a few changes with new research being published after compilation of this list. As we can see, the key on the right explains the different colors, with red indicating more than 10 studies, yellow less than 10, green 2 studies, and black only 1 study available. So the map on the left displays the amount of literature available on human leptospirosis, with Tanzania in the lead with 13 studies, South Africa 4 studies, the rest less than 2 or no studies at all. The map on the right shows the amount of literature available on animal leptospirosis, with most countries at least publishing 1 study, some 2, Madagascar 3, and Tanzania 7. South Africa had the most published data on animal leptospirosis at this stage, 10. This was included purely to emphasize the lack of research available on such an important disease within Africa and why these types of studies are so important. So jumping into my study, this study was based in the Mnisi communal area in Bushbuckridge, Mpumalanga. This area is a regular focus of studies on zoonotic diseases due to its position at the wildlife-livestock-human interface. This area is rural, with 75% of the area bordered by conservation areas such as Manyaleti and Andover game reserves, as we can see on this map on the left. The members of this community are still largely reliant on the environment and make use of subsistence farming. There are large herds of cattle, various smaller rural pig farms, and many household dogs. This community also makes use of croplands and vegetable gardens for food production. Due to the rural nature of the community, healthcare is limited and can be difficult to acquire. One study included in this review included adults from the Bushbuckridge area in a study on various zoonotic diseases. In this study, 6.8% of patients presenting with febrile disease at the local clinic tested positive for leptospirosis. The researchers expanded the study and also found 21.9% of people in close contact, such as cattle herders and veterinarians, tested positive for previous exposure to leptospirosis. The last point to raise would be that a study by the NICD in 2018 identified 3 positive cases of leptospirosis in Valverdinht A. Leptospirosis is normally a sporadic disease, therefore this was considered an outbreak and further investigation was started. The NICD collected samples from various household members, goats, dogs, and water samples, but all of these samples tested negative and the source of the outbreak was not identified. The aim of this study was to investigate rodents as a potential source of pathogenic Leptospira infection within the Mnisi community, where a previous outbreak of human leptospirosis had occurred. As an extension of the study, soil and water samples were also tested to investigate the environment as a potential source of infection. In the case of positive samples, these could be visually depicted by the use of GPS coordinates and geographic images to help identify the possible spread via communal water sources, rivers, etc. This was included after reading an interesting study in South Africa on the spread of leptospirosis within multiple farming units due to effluent water. Moving on to a brief description of the methodology. The sample size calculation for rodents as well as environmental samples were calculated using this formula shown here. For both these sample populations, a precision of 50% or rather 0.5 was used, as the prevalence of leptospirosis in rodents as well as environments is unknown, and we wanted to maximize the sample size. A level of confidence of 95% was used for both of these calculations. A total of 202 rodents and 49 environmental samples were collected, with a simple random cross-sectional study was performed. 34 rats were collected from households in the Valverdinht A. These rodents were trapped overnight using traps as shown in this photograph. Picture A indicates a tomahawk trap and picture B shows a Sherman trap. 8 traps were placed in each of the 25 households. The traps were set in the late afternoon, baited with peanut butter and oats, left overnight, and checked in the early mornings, for a total of 4 trapping nights. Additional samples from the biobank, collected for various other projects over the past 2 years, were also included in this study. These rodents were collected from Atoll, Clavakisa, Gothenburg, and Utah. Soil and water samples were collected from all of the villages. These samples were collected from muddy areas, shallow water bodies, dip tank areas, drainage lines, and areas where there was a lot of interaction between the members of the public and their livestock. Rats were euthanized using a cotton wool swab saturated with isoflurane and labeled with the collection date, the number of rats, and the collection address. These rats were dissected using a biosafety cabinet and their kidneys removed for DNA extraction. Environmental samples were transported back to the lab, refrigerated, and the DNA extraction performed as described in the next slide. So for rodent kidneys, DNA extractions were performed using the Invitrogen PureLink Genomic DNA Minikit, And for both water and soil, the ZR Soil Microbe DNA Microprep kit was used. The extracted DNA was prepared and transported to Onderstepoort for further analysis. A real-time PCR was performed on all the samples using the TACMAN probe and targeting the LIPL32 gene as described by Gentilini with some modifications. Optimal conditions were identified by making use of 5 positive samples provided by the NICD. All of this information was entered into Excel tables and plotted on Google Maps for further analysis. This table is a brief overview of the number of samples collected. As we can see, 34 fresh rodent kidneys and 124 samples from the biobank was included. These biobank samples were collected between October 2020 and March 2022. 25 water and 25 soil samples were included in this study, 5 from each of the 5 villages. As we can see, none of these samples tested positive for pathogenic Leptospira. So this slide shows the proportion of samples from each village to show the distribution between the villages. The different villages are circled on the map to the right. So Gothenburg had the lowest representation as there was community unrest during the time that these samples were collected. I've included a few slides with some pictures. So this slide shows an example of a household where no rats were trapped and a household where 8 rats were trapped. As we can see, the picture on the left shows large amounts of forage easily available, which meant that the rats were less inclined to enter the baited traps. The picture on the right shows an outdoor kitchen area where no forage is freely available and the food is covered or lifted off the floor. This meant that there were fewer alternative food sources available and rodents were more likely to enter the baited traps. For the environmental samples, a description of each of the samples was acquired, along with the GPS coordinates and a photo. Each of these coordinates were plotted using Google Maps and can be seen on the image on the left. The terrain was included here as it shows the location of bigger water bodies, housing areas, and road infrastructure. The pictures on the right are 2 examples of environmental sample areas. The top image shows a drainage line where water and soil was collected. If you look closely, you can see various animal tracks moving through this area and drainage water from various areas in the community. The bottom image shows another side where you can visualize the crush and the dip tank in the background, where large amounts of cattle, herders, and even dogs move through on a regular basis. You can also see the trough with stagnant water, which these animals share when moving through the dip tank. Although rodents have primarily been researched in the past alongside other species, the dominant reservoir in pathogenic leptospirosis is still mostly unknown. In this study, a representative sample was obtained, including rodents from multiple households where they had close contact with livestock, dogs, and humans. These rats were from areas where possible urine contamination of food or water could have occurred. The environmental samples were from high-risk areas such as communal water sources, vegetable gardens, drainage lines, dip tanks, and kraals, where livestock are regularly present and humans could potentially be infected. The environmental samples were also collected after a period of high rainfall, with large amounts of standing water present throughout the villages. All rodent kidney samples and environmental samples had no pathogenic Leptospira bacteria in high enough concentration for the PCR to detect. An interesting observation was that another student used these same rodent samples and performed a PCR using the cybergreen assay, and some of her samples have tested positive. The melting curves produced have been similar to the positive control samples provided by the NICD. There are still lots of questions to be answered regarding the relevance of this, and her study is still ongoing. One may wonder whether her samples are testing positive for pathogenic or non-pathogenic bacteria, and therefore what the implication would be for the community. I think a valuable extension of this study would be the inclusion of more species, such as cattle, pigs, and dogs, along with larger sample size of rodents and environmental samples. One thing to consider in this scenario would be the use of Leptospira vaccination in dogs, and therefore the interference with certain testing methods. Some limitations of this study included a relatively small sample size due to limits in trapping time and the funding available for this project. The original objective of this study was to determine the prevalence of Leptospira within the Mnisi community, and the sample size calculated as 202 samples. Due to the limited trapping success within Valverdine A, only 34 rats were collected, therefore bringing the total rodent kidney samples analyzed to 158. Since all of the samples tested negative, one would be inclined to say that there is no Leptospira present within the study population. This, however, might not be true as the sample size was not large enough to certify freedom of disease. In order to prove freedom of disease in very large populations, such as the rodent population within the Mnisi community, one would need 332 samples, calculated by taking the design prevalence, unit sensitivity, and required population sensitivity into account. This is obviously a much larger sample size than what was required for my study. A more focused approach to the Valverdine A as the primary area where the positive cases originated from in 2018 may be more beneficial and something to consider for ongoing research. With a large enough sample size, one can certify freedom of disease within this village. The apparent absence of pathogenic Leptospira in this study may be disheartening. However, due to the overall lack of available research on leptospirosis in South Africa, any information is still valuable. This study encountered many hurdles along the way to completion, which emphasized the difficulty of pioneering research on a disease due to the overall lack of resources, the lack of knowledge regarding the perfect testing methods, and the unavailability of the tests themselves. There is still a long way to go in order to learn about and understand leptospirosis within South Africa, and it remains important to strive for a better understanding, as so many communities are still reliant on the resources of the land. These communities make use of communal water sources, rivers for bathing, self-cultivating vegetables, and livestock farming. Without proper education and prevention of disease, these practices could be putting members of the community at risk. As a final remark, even though this study didn't make any groundbreaking discoveries, it serves as a building block for further research, and it raises awareness within the scientific community. Thank you very much. I hope that wasn't too much and too long.
Thank you, Angelique. I know you had a lot of roadblocks post-COVID to complete your study and practical work. I have a question for you. In the environmental samples, do you think the time of the year or season mattered, that it would change the results if you did included more seasons?
I think we ended up including samples because we used such a big sample size through various different years and therefore various different seasons. I do think that we included a nice, well-rounded sample size, and I think we did include some rodents from each of those seasons. However, I was under the impression that we would potentially get a higher amount of Leptospira positive cases by going in the rainy season, where you're seeing all of these puddles of water standing around. You have all of these rivers overflowing, lots of movement of water, which is obviously your highest risk with your or hypothesized highest risk of your leptospirosis. Although we didn't have any positive samples, so whether it is relevant or not would be difficult to know. But there are some studies that suggest that water definitely plays a role. And there's some studies that suggest that human leptospirosis increases in the warmer seasons due to the increased amount of water sports being practiced, people being in the water more, swimming, boating, all that sort of stuff. So it could definitely play a role.
Perhaps during the dry season, animals congregate more at water. That remains to be seen. Another question, what about vaccination in the community? Do you know of vaccinations being done in dogs, for instance, or do you think that influenced the results of your study?
So in that community and throughout some areas of South Africa, there are community service veterinarians, so CCS, who's performing their community service. And some of those clinics do have access to some of the lepto vaccinations. So those are government-funded clinics, and a lot of them don't always have lots of vaccinations. Some clinics have different vaccinations to others. So there's definitely some areas of South Africa that are vaccinating against leptospirosis, but I do suspect that it's very much a how much stock you have and what you have available at your clinic specifically. It's definitely something to take into consideration if we are, or if there's further research being done on leptospirosis, specifically testing in dogs, since you do need to just take that into account if they are lepto-vaccinated, and what the implication would be of that. But obviously for my study, it wasn't overly relevant. It's more relevant for ongoing research.
Sorry, I was on mute. Thanks for answering that. I don't see any questions in the Q&A or in the chat box. Anybody have something they want to ask? Just for interest, the parallel study that you mentioned, I just want to check you can hear me.
Yes, I can.
There's nobody else answering. There's a question from Elizabeth. Hello, thanks for the interesting presentations. Any suggested behavioral science interventions?
Do you maybe just want to clarify on what you mean with behavioral science interventions?
Elizabeth, oh, there we go. I'm asking in relation to medical anthropology, anthropology.
I suspect that's slightly out of my field. If you maybe just want to clarify what exactly you're asking, then I'm more than happy to give a little bit more information.
I know the people on the outbreak in Valferdine, it was all women. And I know it's the women's role to collect water and basically use water washing. And maybe that is a gender associated because of gender roles in the community.
And I guess in that same breath, you've also got the opposite with your males being more inclined to herd cattle and do the dip tank activities. It is a little bit more old-fashioned in those communities.
Yeah, that is true. In a previous surveillance study by the NICD, it was actually a cross-section in herders, and it was actually quite a number of herders that tested seropositive without clinical symptoms for lepto. Okay, Elizabeth says yes, that it is as well the community perceptions for interventions intended to limit lepto. Thanks.
Yes, I think unfortunately in some of those areas, it's so underdiagnosed that a lot of the community are not even aware that lepto is around or exists. And I think that really makes the intervention for further prevention of the disease quite difficult, because if there's no understanding of what exactly it is or what the risk factor is, or that you need to be preventing it, it makes it extremely difficult to obviously drive these prevention projects.
Yeah, and I also think it's a capacity to diagnose. The symptoms can be quite vague, unspecific. The capacity to diagnose it at the clinic is also limited.
Yes, and in some of the research that's previously been done, it's not even coming across as much of a differential diagnosis because malaria is so prevalent in that area. It's just, if you present with febrile illness to the clinic, it kind of automatically gets assumed to be malaria until proven otherwise. So it's not just obviously the unavailability of tests, but once again that perception that it's not really there, or if it is, it's not much of an issue.
Okay. So just to add additional information, the study you referred to, the student that did the parallel testing, she actually used a different DNA extraction technique. She used the QAGen, and she only found, I think, 3 water samples and 1 soil sample. So she used your samples, and we took additional water and soil samples. So I mean, even with her testing, the prevalence in the environment from the samples we got was very, very low.
Yes, it's going to be interesting if she does write it up to find the better testing method, and just determining which one is a little bit more sensitivity.
Yeah, she used the same primers as you for the cyber green. So I would assume they were the pathogenic ones. Any other questions? Somebody wants to ask something or mention something? No, then I guess thank you, Angelique. It was such a pleasure to work with you. Oh, wait, Clara's positive samples at different melting points, suggesting they were not pathogenic ones. Okay, thanks for clarifying that, Nicola. Okay, well, I guess there's no more questions. Then, Charlotte, if that's all right, we just say thank you, Angelique, and congratulations on your nomination and job well done. And I hope you a very prosperous future and come back for your PhD.
Thank you so much. Appreciate it.
Thank you very much to all of you, to Angelique as well, for sharing indeed your research findings. Also to Ilana for moderating this webinar. Sorry to the participants because the screens are black, but it's due to internet connection. So just to avoid losing the speakers. In fact, also thank you to Mika for being present, and also, of course, to you participants for being present. I will now launch the poll. If you could complete, that would be we would be very grateful. And we would also like to invite you to the 4th and last webinar, which will take place on the 16th of November with MPH alumna Blessing Umegbo from Nigeria. It will be announced on all social media channels and of course the ITM online alumni platform. So thank you once more. Indeed, all the best in your future career, Angelique, and thank you once more to all for being present at the webinar. And have a nice evening still, because it's already late. So thank you very much.
Thank you so much.
Charlotte and Mika, also for all your arrangements.
Thank you,
With-
Ilana.
With pleasure. Thank you very much.
Right, then I guess we say goodbye.
We do. Yes. Thank you.
All right. Bye, everyone.
Bye-bye.
Thank you,
Bye.
Angelique. Bye.